already doing that. It seems like just a reminder to please put any questions or comments that you have into the chat. A lot of folks are already doing that, and that's wonderful. I know our moderators will work hard to try to answer those questions. Also, please be reminded that slides and recordings will be available in about 3 weeks, and you'll be notified of that via email. Before we start into session one, we do want to acknowledge the efforts of today's session moderators, Priyanka Ghosh, Sam Raney, and Tanaz Ramazanli, who are moderating sessions 1, 2, and 3 respectively. Sam, you've already met. Tanaz will be moderating session 3 late this afternoon. She is a senior pharmacologist within the Office of Research and Standards at OGD. She specializes in topical and transdermal products. She's a senior reviewer for the development of product-specific BE guidance, responding to controlled correspondences, citizen petitions, and pre-ANDA meeting packages. She leads several regulatory science research initiatives related to the development of BE standards for complex topical drug products through FDA-funded collaborations with research institutions around the world. Kicking us off this morning for session 1 is the moderator, Priyanka Ghosh. She is a senior pharmacologist within the Division of Therapeutic Performance. Her areas of expertise include products in the topical and transdermal drug delivery areas. She is co-chair of the BE Standards for Topicals Committee within OGD. She leads regulatory science research initiatives related to topical and transdermal drug products, including projects related to the development of non-invasive imaging techniques for the evaluation of cutaneous PK under GDUFA regulatory science program. Thanks for putting session 1 together, Dr. Ghosh.
Thank you so much, Dr. Pauly. As Dr. Pauly mentioned, I'm Priyanka Ghosh. I am the team lead for the topical products team within the Office of Research and Standards, Office of Generic Drugs. And I just wanted to give a brief overview of session 1 before we get started with very exciting talks. So as summarized during the previous 2 talks this morning, the GDUFA Regulatory Science and Research Program has facilitated the development of efficient characterization-based approaches for demonstration of BE of prospective topical products, but at this time, their utility is limited to instances when the formulation composition of the prospective generic product is well matched to that of the reference product. The purpose of the workshop today is to discuss how do we develop efficient science-based BE approaches that may be helpful for certain prospective generic products that are compositionally different compared to the reference standard. These approaches may utilize sophisticated techniques that have been developed to characterize the physical, structural, sensorial, and thermodynamic properties of topical drug products applied to the skin, among other techniques such as the cutaneous PK techniques that will be discussed in the 2 later sessions during the day today. So for this session, the session 1 of this workshop, we will focus first on understanding the current challenges with demonstrating bioequivalence efficiently for prospective generic topical drug products that may have formulation differences compared to the reference standard. And when we are talking about formulation differences, we mean that the prospective product may not meet the no significant difference criterion compared to the reference standard that is often outlined in our product-specific guidances. We would really like to understand what the challenges are for the industry in this space, and to be able to identify what kind of research may be needed to resolve such challenges. And we are very much looking forward to hearing from our industry speakers during this session today. During the second half of this session, we will discuss new techniques that are being developed to characterize the quality and performance of topical products, including sensorial and thermodynamic considerations, to understand how formulation differences may influence the Q3 characteristics of the product, and may also influence bioavailability and therapeutic equivalence. So without further ado, it is my pleasure to introduce the first speaker for this session. I'll note that in the interest of time, as Dr. Pauly mentioned this morning, we will be using abbreviated biographical information for all speakers for these introductions. However, the full bios are available on the CRCG website. Our first speaker today is Dr. Sachdeva. Dr. Sachdeva is an associate director for drug product development at Amneal Pharmaceuticals. His expertise includes topical and transdermal drug delivery systems. He holds a degree, BS degree in pharmacy from the University of Delhi and a PhD in pharmaceutical sciences from the University of Pacific. Dr. Sachdeva has published numerous research manuscripts and is also on the reviewer board and editorial board of numerous peer-reviewed journals. Please join me in welcoming Dr. Sachdeva.
Thank you, Priyanka, for a wonderful introduction. Hello, everybody. My name is Samir, and today I will be talking about the effect of formulation differences on the critical quality attributes and performance of the complex topical products. So I'll be giving a brief outline here. I'll explain the concepts of Q1, Q2, Q3, and then the arrangement of matter Q3 and its importance, and discuss a little bit about the non-Q1, Q2 formulation or the formulations which have significant differences that will cause the product complexity. A few product development approaches, how can we approach these kind of formulations, and studies to demonstrate BE for these products. So here are the concepts of Q1, Q2, Q3. I think most of us are aware of these concepts. Q1 refers to the components or the qualitative composition, which include the chemistry and grade of each inactive ingredient in the formulation. The Q2 refers to the concentration in that of particular components. That is the quantitative composition. And for sameness, the difference should not be more or less than 5% of the reference product. Whereas Q3 is the physical, chemical, and structural attributes, which is also we refer to as arrangement of matter and includes a lot of different parameters or attributes such as pH, globule size, particle size distribution, neurological behavior, drug polymorphic form, etc. So I'll talk more about these Q3 parameters in the later slides. Let's go over what considers these sameness, similarity, and Q3 differences. As Sam referred that there's a draft guidance came out recently, which defines all these three different characteristics. So for Q3 sameness, Q1, which is the component, should be same as the reference product, same composition within the acceptable differences, and same or similar Q3 parameters, which will fall within the variability of the reference product. Where Q3 similarity is discussed where Q1, Q2 may not be same. There may be differences in the components on composition, but the Q3 properties will be similar. And when I say similar, it should be within the variability of the reference product. Q3 differences where the Q1, Q2 of the formulation may be same or may be different, but Q3 properties are different. That falls in this category. So here are the Q3 parameters or attributes which are looked at for the complex topical product. First is the appearance and texture, which gives the look and feel of the product. How does the product feel? What is the odor of the product? And this defines the description of the product. Phase states and organization of the matter that describes how does the product look like under the microscope? Are there more multiple phases or single phase? What is the particle size? What is the globule size? Just to define the whole dosage form. These first three characteristics come under the basic Q3 characterization. Then the last seven comes under the comprehensive characterization. For example, drug polymorphic form. Different polymorphic forms have different stability, different solubility. So this is very important to characterize the polymorphic form in the product. Rheological behavior is one of the major Q3 attributes which should be looked at while characterizing Q3 parameters. Is the viscosity same? Doing at different shear rates? How does that differ when the process is changed? The next is water activity or drying rate. How does the formulation versus the reference product dries out? If it has alcoholic or hydroalcoholic behavior, then how fast that dries up. And that changes the whole product performance based on the thermodynamic reactivity of the API. pH and buffering capacity. So pH should be very critical as it can change the ionized solubility of the API. Oleaginous component, this is more for the petrolatum based components where hydrocarbon characterization is important. Specific gravity, just to know that how much air is in the product, and when you dose the product, how much API we get out of the product. Metamorphosis changes. This is really critical as to know how does the product comes out in the different packaging components, for example, from tubes or pumps. And then overstability, are there any characteristics Q3 attributes are changing? For example, if the product ages, is the particle size changing? Is the globule size changing? And this can be done actually doing the reference product, aged reference product versus the test reference product, test product, just to see how are the changes during the age of the product. So as I discussed, Q3 parameters or attributes of the reference product demonstrate the pharmaceutical and bioequivalence position, and it can mitigate the risk of the potential failure modes if we are matching that with the RLD of the reference product. And it can help us mitigate the risk in the changes in Q and Q2. If there are changes and Q3 parameters are same, you are very well and may not have and could avoid these potential failure modes. pH, that can irritate the skin, and if you have the same pH, that would avoid that. Again, I discussed the polymorphic form, rheological behavior, and the other component is the partitioning of the drug, the diffusion of the drug. If the Q3 parameters are same, globule size, where the drug reside, location of the drug, if it's a multi-component system, that can really match the drug release of the test product versus the reference product, and drying rate and metamorphosis. So if the drying rate and metamorphosis of the test product is similar to the reference product, it really shows that the product performance could be very similar to the reference product. Let's talk about the product complexity. And we know that if the product having the same Q1, Q2 or qualitative and quantitative composition, that could result in different appearing and functioning products. Saying that a Q1, Q2 formulation can have different Q3 attributes, and that can have a different product performance. And a test product with same Q3 attributes as a reference product can actually prevent the known risk failure modes. As I think our peers mentioned, generic topical products may not be Q1, Q2. And there are enough data to demonstrate that the products which are not Q1, Q2 or Q3 same, they are bioequivalent based on, let's say, in vivo clinical studies or other studies. So what are the situations that may lead to non-Q1, Q2 or different formulations? It could be very difficult to reverse engineer. It has a one particular, let's say, component is a multi-component system, and it's very hard to reverse engineer that. Or there may be intellectual property issues that you can't use the same component in the same percentage, or the RLD is discontinued and we don't have the access to it to reverse engineer. And the reference standard, let's say, may have a different profile altogether. So let's discuss about a product development approach of these kind of formulations. So when a product doesn't qualify for this characterization in vitro approaches, maybe non-Q1, Q2, it's very important to understand the reference product in the respect of composition. What is the role of each inactive ingredient? And if there's a change, how does that impact the CQA? Grade or chemistry of each ingredient is very important. And knowing that if we change that, how the CQA is impacted and how does that impact the product performance? The phase state and all the arrangement of matter, is it same though this non-Q1, Q2? And how will impact the rest of the critical quality attributes? Drug diffusion within the dosage form and how it impacts the critical quality attributes. It's also important to understand the process. A process could have significant importance in this complex product for topical dosage forms. The sequence of mixing, the speeds, the duration, temperature, all this can result in very different Q3 attributes, even if you are Q1, Q2. So it's very important to understand the process and trying to match the process to get the similar properties as the reference product. The question is, if we are still non-Q in Q2, or there are differences, can you still have the Q3 similarity as the reference product? The goal should be to achieve the Q3 similarity. As I discussed, Q3 similar formulation will avoid the risk of known failure modes and will have pretty much similar product performance. The goal should be to match Q3 attributes and CQs as close to reference product for especially non-Q and Q2 formulation, characterizing the reference product extensively and see the changes in the test product and how does that impact the failure modes. For example, purposely changing the percent of ingredient and testing the failure modes. Does the difference, let's say if there's a polymer difference or the surfactant difference, how does the change affect the CQAs and Q3 properties? And how does that change in the Q3 properties alter the product performance? This is an example from Jani and et al. This paper was published recently in 2022, in pharmaceutics. So if you see, these are different formulation for diclofenac sodium. First is the emulsion. I'm not going to go in detail with the composition. For the next is emulgel. The difference is just one of the polymers, Clucel. Third one is the gel phase, where there are not a lot of ingredients of lipid ingredients. And the last is the ointment, which has polyethylene glycol-based ointment. If you see the loss modulus and the storage modulus here, it shows that the lower viscosity is for the diclogel, and the emulgel and the emulsion are in between, and the highest viscosity is found for the ointment. So these are the Q3 parameters, and they are different for each of the particular formulation. And if you see, this is the IVPT data. The IVPT data shows that the emulgel, which is here where there was one extra component, Clucel, it has a higher permeation rate as compared to the other formulations. And the ointment base had limited or negligible permeation. If you see the last plot here, it clearly shows that in the receptor media, they were not able to find any API in the ointment, and the emulgel formulation had the highest amount from the receptor and more of the permeation through that skin. Similarly, tape strips method show that most of the API from the ointment is on the skin, in the stratum corneum of the top layers. And in the epidermis, the emulgel had more of the API because of the higher permeation rate across the skin. So this shows us that if there are changes in the formulation and Q3 properties are different, it will result in completely different performance of the product. So here are the BE approaches. I think Sam covered some of these. So characterization-based approach, in vitro-based approach, where we have Q and Q2 sameness, Q3 sameness, and then doing IVRT, release of the product from a membrane, and IVPT, release from the skin. If these are for same Q and Q2 sameness. Alternative approaches are clinical endpoint study or in vivo, let's say vasoconstrictor study. The issues with these studies are that these are very time-consuming, expensive studies, and there are a lot of variability, need a lot of subjects. So it's a better option to go with in vitro technique if that's possible. And pharmacokinetic study, if there's enough API in the systemic circulation, we can do a PK study, in vivo PK studies to show the amount of drug in the systemic circulation. And in vivo cutaneous PK studies. There are non-invasive studies such as confocal or simulated Raman spectroscopy, and the other are dermal microlysis or dermal open flow microperfusion. I think these will be discussed in the later sessions today. So let's go over and discuss how confocal Raman spectroscopy can be used. So this is the paper by Ilopoulos et al. So in this case, there were two different diclofenac formulations. These are EU-based formulation, diclac lipogel and primofenac emulsion gel. If you see the IVPT study through the skin, you can see that the diclac gel has 3-fold higher permeation from the skin as compared to the primofenac. And very similar data, which is correlating with the IVPT study, was found in the confocal Raman spectroscopy. If you see, this was done, and then approximately 3-fold difference was seen which was similar to IVPT studies. And when they did the tape stripping study, it was found that for the diclite gel, most of that drug was present in the skin as compared to the pimofenac. So this data shows that the confocal Raman spectroscopy can be used and can show the results, which are very similar to the in vitro permeation studies. So I'm going to go over with the 3 different scenarios. Let's say if the Q1, Q2 of the formulation is same, and you will be able to achieve the Q3 sameness, that would lead to the characterization in vitro BE approach, let's say IVRT, IVPT. In the second scenario, if the formulations have differences or they're non-Q1, Q2, but if you're able to match the Q3 properties and Q3 properties are similar, then in this case, you have to identify and do the extensive characterization for the failure mode. For example, if you know that there is a change in one of the components or one of the components is different in percentage, what are the failure modes of that? Extensively changing that and seeing if that affects the product performance or not. Do IVRT studies, IVPT studies, additional studies, as I discussed, will give you the in vivo cutaneous data, CRS, SRS, and microdialysis or the microperfusion studies. And this will be able to show that if the failure, what are the failure modes, and are the failure modes mitigated, though we are using a different formulation, but is the product performance safe? And the third scenario is where it doesn't really matter if it's Q1, Q2 same or non-Q1, Q2. If the Q3 properties are different, they have to go to an in vivo clinical endpoint study or other PD studies. To conclude, for a topical complex product, it's very important to do a thorough characterization of components, compositions, and Q3 properties, physical and structural characterization. Characterization-based approach is possible when the Q2, Q1, Q3 are matching. And Q3 characterization of a reference product is important, and that can actually mitigate the known risk failures. And if there's no non-Q1, Q2 formulation, multiple studies can be conducted in vitro, in vivo, other cutaneous studies to show the product performance if Q3 similarity is there. Thank you.
Okay. Thank you so much, Dr. Sachdeva, for the wonderful presentation. We will move on now to the second presentation that we have today from Dr. Vaibhav Dubey. Dr. Dubey is a Deputy General Manager for Specialty Formulations at Alembic Pharmaceuticals in India. He was previously associated in different roles with Sun Pharma Advanced Research Company, Sun Pharma Industries Limited, and Dr. Reddy's Laboratories. He has been instrumental in developing numerous topical generic products and novel drug products intended for filing using the various routes, various regulatory pathways that are available within the FDA, and has contributed immensely to development of bioequivalent strategies centered on the in vitro characterization-based approaches that were discussed earlier today. Dr. Dubey has almost 2 decades of research experience in development and characterization of topical and transdermal drug products, and several publications and patents to his credit. He has a Master of Pharmacology and PhD, where he studied skin permeation mechanisms for drug-laden modified liposomes. So, Dr. Dubey, without further ado, would you be able to bring up your slides and we can get started with your presentation? Thank you.
Thank you, Priyanka. Thanks for a kind introduction. So we'll start with the presentation. So I will be presenting on the development strategies for generic topical products with formulation reference to reference product or reference listed drug. So,
Dr. Dubey, we still see your slides, not the presentation screen.
Is it
Yes.
visible now?
Yes, thank you.
Right. So I will be presenting on the development strategies. There was a nice presentation by Samir. So he has already defined Q1, Q2, Q3. He also defined the new guidance, what FDA had recently published on Q3, how Q3 same, similar, and different products you can look at. So my presentation is just an add-on to Samir's presentation. Now, I would be discussing more from an industrial perspective, that when we add a product into a development portfolio, we look at certain different aspects of that product, that whether what's the indication of that product, what's the characteristic of that product. It's a solution, cream, or a complex topical product, or microsphere one. What's the current IP scenario for that product? Whether deformulation is feasible in that product or not, or what's the site of application, drug availability of that product, whether there is a product specific guidance available for that product, how many, what would be the possible volume of that product. There would be a once a year batch, manufacturing batch, or there would be 10 batches a year, a month for that product. How many prescriptions are actually dermatologists prescribing for the innovative product for such for that, and how we will be placing our generic product, how many generic players are there for that product. So all these things have to be kept in mind prior to deciding that we should be having a formulation similar to reference product, or we can have a formulation which is different to the reference product, and then we can invest more on a bioequivalence approach, which is generally a clinical endpoint approach, and we can invest more, and then we can look at the cost actually for the development cost for that product. So once we had defined the product in the defined phase, actually, we go into product development. So with the deformulation feasibility, we are pretty sure that, okay, we can have a formulation similar or different to the reference product. If we have a formulation similar to the reference product, we know that, okay, in vitro characterization based approaches would be there actually. So if it's a Q1, Q2, Q3 product, so we know that there are several published product specific guidances, or if they are not, if we can approach FDA with a pre-NDA package. And mostly in most of our cases, we found that FDA was too helpful actually to come up with this in vitro based characterization based approach for such products, or if there are other relevant bioequivalence approaches for such products. In cases where formulation differences are there to the reference product, we take a strategy that we either approach FDA with Q1, Q2 proposal, multiple Q1, Q2 proposals in that particular pre-NDA package, and look for FDA's opinion that, okay, with this composition, with this type of product, whether a Q1, Q2 approach is feasible. And then we also provide the Q3 data in that pre-NDA package. And we suggest from our side that, okay, this could be a relevant bioequivalence approach for this particular product, which is different to the reference product. FDA agrees, then we can look for a relevant bioequivalence approach. If FDA doesn't agree, and most of the cases in this scenario, there are less likeliness that FDA would agree. So we look for a clinical endpoint based or a relevant bioequivalence approach in that case, and post that we go for an ANDA filing. So in the defined phase, actually, as Samir has also mentioned, we'll define the product, what we need to develop. And for defining the product, what we need to develop, we need to define the RLD or the reference listed drug. So we do a deformulation as well as a Q3 characterization of multiple lots of RLD. So as to establish a range of for Q3, actually, we cannot have one viscosity value actually, or one rheological profile for RLD, one batch of RLD. We had seen several cases wherein there are difference in RLD lots itself. So we should have a range of RLD Q3 values as well as the other parameters for RLD lots. We study them in quite detail. We also looked at whether this PSG or a product specific guidance is available, what type of approaches it has been mentioned in those guidances, like it's an in vitro approach or a in vivo approach. Can we still look for a in vitro approach wherein product specific guidances are available? Still we have a concept that FDA may agree for an in vitro based approach for a product specific guidance, which currently mentions a clinical endpoint study. We also assess the current IP scenario when actually we can come to the market with that particular product, how like ease of deformulation, nature of bioequivalence study, like suppose it's an acyclovir cream, I'll quote an example. We cannot look for a clinical bioequivalence study because it's really a huge study with 4,000, 5,000 patients and almost 4 or 5 years. So in such a scenario, we have to be have to make a formulation which is Q1, Q2, Q3 to the RLD. As Samir also mentioned that for formulation for products wherein we look for formulation differences, is somewhere where IP scenario or intellectual property scenario is not clear at that moment. Deformulation is not feasible. There is there is non-availability of some specific component which is being used by reference product. Maybe it's because of IP, or they have some specific marketing authorization with the excipient company, or in certain cases wherein reference listed drug is discontinued, or there is a lack of clarity on the reference standard. So once we have defined this in the define phase, we go to formulation development. In the formulation development stage, we will define CQAs, CPPs, CMS, and everything. And when we look at, and when we find that, okay, there are differences in components or compositions actually. So in certain scenarios wherein suppose I have a solution product and I am looking for a change of antioxidant, suppose a change from BHT to BHA. BHT is also soluble in that solution. BHA is also soluble in that solution. So for me, that won't be a critical thing because it won't be impacting my Q3, or it won't be impacting my bioequivalence also. So for me, the critical component in that scenario may be a penetration enhancer. Suppose in case of gel, I'll look into a critical component that could be a gelling agent. It could be a pH independent gelling agent or a pH dependent gelling agent. So in certain scenarios, we have to define that what would be critical actually for that particular formulation type. So when we define it that, okay, this thing is critical, then we have to define the change, up to what extent we are making a change, whether it shall impact, what it will impact, whether it will impact the comparative Q3, or it will impact our relative bioavailability or bioequivalence. And what could be my risk mitigation strategy? Suppose I'd say that, okay, the changes, the critical component change is happening and it is significant. So I may be looking for a clinical endpoint study. Now to de-risk that clinical endpoint study, because there is a huge cost involved there to do that study as well as time. So we have to look into that, what failure modes we should look into in these products that saves our time and energy actually in doing this clinical endpoint study. So I'll just present one case study here. It's a known product actually. I have just kept it this way, that okay, I have a formulation, it's a topical gel product wherein the API is intended to be available within the skin milieu. It's an acne product, so obviously it needs to be availed, the API or the drug needs to be availed at the pilosebaceous units. So I have a RLD product which has an API, it has a penetration enhancer transcutol. It consists of a pH independent gelling agent. This pH independent gelling agent consists of a polymer, it consists of a dispersion media and a surfactant. Then RLD consists of preservative and purified water. Suppose if I go with the Q1, Q2 approach, I will be having the same composition, same components and composition to RLD. However, if we take a different approach wherein we are changing a pH, the gelling agent. So from a pH independent gelling agent, if we want to move to a pH dependent gelling agent, so with that change actually, there will be a change in the critical excipient, that is a gelling agent. Now, if I am adding a pH dependent gelling agent, I need to add a neutralizer or a pH neutralizer like sodium hydroxide into that case. What I can do is I can match the surfactant content, what is already there in the pH independent gelling agent in this case also. However, this particular composition will be quite different to that of RLD. Now, in one case, I am adding a neutralizer. In the other case, I am not adding a neutralizer. So in this case, I'll say that Q3 similarity would be highly challenging. I'll say that this will be a Q3 different products altogether. There would be multiple aspects, multiple Q3 attributes that would be different in of this generic product with different Q1 to that of reference listed drug. So in this case, what we do, as Sam also mentioned, that we'll be looking at the failure modes. I'll say that we have to look at relative similarity of the Q3 attributes of this generic product, what we have made, to that of RLD, so that we can target the risk mitigation to do a bioequivalence study. Going to the next slide. So this approach just says that, okay, if I have a generic Q1, Q2 approach, wherein the arrangement of matter or underlying matter remains same, I have an approach, a bioequivalence approach, which says that, okay, I can do an IV, RT, IV, PT or PK. However, in case wherein I have an approach wherein generic product is having a different Q1, Q2 to that of reference listed drug, in that case, the arrangement of matter would be different entirely. So in those scenarios, I can have a possible difference in rheological features of both the products, appearance, texture, specific gravity, pH, and a dissolved to undissolved ratio. So how to mitigate it actually once we have these things different? So we can look for a range of dissolved to undissolved drug content actually that would be impacting the clinical endpoint study. Now remember in this case or the other cases wherein we have an acne or a psoriasis product, the clinical studies are for 12 weeks. So those are long studies. So obviously they are subjective in nature, but in terms of drug availability at the site, if you see, it's a multiple application study. So there would be a daily once or twice application. So skin saturation can happen better actually in case of a long study as compared to doing an IVPT study with a single dose. So that scenario also is there. So that subjectivity lies with the clinical endpoint studies, but chances of success are also bright actually as compared to that of a single dose IVPT study. So either you can look, go with a Q1, Q2 approach, Q1, Q2, Q3 approach and do a clinical endpoint study if IVPT is not getting successful because of variability or any other stuff. Or if you have a different product altogether, obviously you have to go with a clinical endpoint study. So coming to the second thought is like if you have same Q1, Q2, but there is a Q3 difference actually. So now Q3 difference may or may not impact your bioequivalence. So, and in that, in one case when it can impact the relative bioavailability or bioequivalence, then you should look into that, okay, the clinical endpoint study, what you would be performing. So where actually you are looking for the PD response actually, whether it's a locally acting drug or whether it has a local plus systemic feature, or like an acne product wherein you need the drug to be retained within the skin actually. Or the type of bioequivalence study you are doing, it's a PK or a cutaneous PK type of stuff, or any other approaches like you are running a pediculicide hair tuft assay. So in that case, with like benzyl alcohol or something of that type. So whether this particular change, Q3 change, would be actually impacting the anti-lyse activity. So that relative thought you have to put in and accordingly design or look for the bioequivalence study. Going to the next slide. So this is just a case study of such type. So this was a case when we developed a product of this type, wherein the product was Q1, Q2. However, we were not finding Q3 same. And what we found was in that case, when we were evaluating different RLD lots of different ages, we were finding certain structures in the RLD lots, which were like the intensity of those structures were actually differing with the ages of the RLD, as well as those structures were only visible in the dark light or in the polarized light, you can say. So we thought, okay, that could be a aging effect actually in the RLD that may be bringing in those structures. So we also designed our product in that scenario that those structures are visible. We also thought of like that we should have a design space wherein we get those structures which look similar to what we are getting in the RLD. However, there was no control what we saw actually in the RLD lots themselves. So saying it as Q3 is different or a Q3 similar, not Q3 same, maybe a point actually that we can make in this particular sense. Going to the next slide. So what we thought is like if RLD is in this case, actually there were two scenarios. One is RLD tend to depict a decreased apparent viscosity during shelf life. So what we thought was like, okay, what we can do is at max is we can have a test an RLD product as closely matched in age as possible. Because obviously when RLD manufactures it and it comes to the market and then we procure it from the market, it's almost 5 months, 5-6 months. We do not know actually what age, what time RLD has actually marketed that product. It's the expiry date that is given there. So we try to match at least that age. And obviously we can do a comparative release testing actually. So if with similar age test product and RLD, we get a similar IVRT, at least we can go for a IVPT with such type of product. The second scenario, what we noticed was, as I was mentioning, was the structure, different structures we were looking in that, but we were finding in that particular cream product. So what we found was that those presence or absence of those structures were minimally impacting the release of the drug. And what we investigated was that those structures were being generated by some structure forming excipient in the cream product, as well as that those along with availability of the surfactant was making those products. So those were coming in placebo also. So we found that, okay, it was minimally impacting. So we can say, rather than Q3 different, those products were Q3 similar, not Q3 same. Then we optimize the process design space, and we could find that, okay, those structures were visible here as well as in the RLD, and we could have a similar product actually. Going to the next slide, it just mentions how what we can do actually for a selection of a bioequivalence approach, what factors actually are impacting with the bioequivalence approach selection during such generic products which have formulation differences. Everyone tries actually to have a in vitro approach, because those are obviously less time consuming and cost intensive. However, in cases wherein the product is actually maybe a first to file product, or you have a CGT opportunity there, you can think of a BE strategy, which may be a clinical endpoint approach, or which may be cost intensive also, but it will try to bring you early in the market actually. Then there could be other factors like number of studies to be performed. If I look from an in vitro bioequivalence based approach, I have to do a Q3, comparative Q3, then IVRT, IVPT, and in certain cases PK also. Whereas doing a clinical endpoint study, there is only one study, though it's subjective, but likeliness of passing the study based on how powered the study is more sometimes. So that you have to weigh in actually which approach is better for that particular company. Then you can look, then there are factors like specific geographical requirements or skill set requirement, like for a DOFM or a dermal open flow microdialysis, or something which is not available everywhere actually. So we have to look for such CROs actually if we want to do it. There are pharmacoeconomic factors also, that certain studies are too costly because they are still under development. Then you can weigh in also that whether you want to go with a well-established technique or a novel method for bioequivalence assessment. And then you can also look at the variability actually. If you have two different approaches available, so which is more variable, and which will yield a better FDA acceptance. So all these things you need to weigh in actually for selecting a bioequivalence approach for such a strategy. Concluding here is like, I'll say that when you define a development strategy for such a product, which has formulation differences to the reference listed drug, all the strategies would be tailor-made based on the type of product indication, site of product application, site of drug availability, relative similarity to RLD, and extent of change that you are making. Then difference in component composition of Q3 should be studied well and correlated to product performance to support bioequivalence. And bioequivalence approach, as per the industry requirement of a particular company, must be time and cost saving for early market entry, as well as lower overall development cost. Thank you.
Thank you so much, Dr. Dubey, for the wonderful presentation. We will now move on to the next presentation in this segment. Our next speaker is Mr. Romit Jani. Dr. Jani is the Director and Head of Formulation R&D at Solaris Pharma, a company that primarily focuses on the development of complex dosage forms. Currently, he leads the research and development activities of the company, including formulation development, scale-ups, and commercialization. Prior to Solaris, he has worked as a research scientist at Amneal Pharmaceuticals and was responsible for formulating and scaling up various complex dosage forms, including topicals, ophthalmics, etc. He received his Master of Science in Pharmaceutical Manufacturing from Stevens Institute of Technology and has around 10 years of experience in the pharmaceutical industry, taking products from concept to commercialization. So please join me in welcoming Dr. Jani for his presentation. Mr. Jani, sorry. Romit, would you be able to bring up your slides?
Yes, hi.
Thank you.
All right. Thank you, Dr. Ghosh, for your kind introduction, and thank you, CRCG, for providing me an opportunity today to present on this forum. Hi, everyone. Today, I'm going to discuss some of the challenges and possible suggestions or solutions for establishing bioequivalence using characterization-based approach for topical products. So as an outline, today I'm going to discuss what are the challenges for establishing Q1 and Q2 sameness characterization that exist for several complex excipients, which are very difficult to reverse engineer. Along with, I'll provide one case study for how changing a particular polymer grade impacted pharmacokinetics. And later, some of the thoughts that we have on non-critical excipients, or when I say non-critical, it's for bioequivalence, and how we can still fall outside 5% and still be bioequivalent with the summary. So next slide. So there are several challenges exist for any topical generic product development. I have listed some of them here, such as RLD lot-to-lot variability that has an impact on Q3, as well as IVRT and IVPT. There are some certain challenges exist during IVPT studies, including some of the statistical outlier thing. Also, when it comes to low dose concentration drugs, there are several challenges exhibit to measure the particle size and globule size. However, today I'm going to discuss more on the Q1 and Q2 related challenges, that how Q2 related challenges, right? How critical is the reverse engineering, and a lack of availability of analytical techniques makes it more difficult for certain Q1, certain complex excipients like petrolatum and carbomers. Also, there are certain challenges exist in Q1 front where there are certain excipient listed in labels, RLD label as a single excipient, as a separate excipient, but when we source that from manufacturer, it is available as a single excipient. So how to determine that, and then ratios of those excipient is very critical as well. Also, I would like to discuss some more in detail about reverse engineering challenges for some complex excipient, and if there are any other approaches available for industry to establish bioequivalence for the products which may fall outside the 5%. In this slide, I would like to explain a little bit about qualitative challenges, sameness challenges. As I explained, there are several scenarios where industry is having an issue establishing qualitative sameness or Q1 sameness for the RLD or reference listed drug. One of them is, as I explained, there are several innovator products which uses a combination of excipients, and it is available as a combination in the market. However, RLD label indicates them as a separate excipients, such as glyceryl stearate, PG100 stearate, sucrose stearate, sucrose distearate, some of the polymers, carbomers. So if you can see on the right side here, I have listed some of the examples of those where industry is facing challenge establishing Q1 sameness in that with that regards. Also, there are several issues that industry is facing during identifying the grades of particular excipients, where RLD label just mentions petrolatum or carbomer. So it is very difficult to identify a particular grade of those excipient in that case, as well as when it comes to carbomer, it is difficult to identify what grade of carbomer that is being utilized to establish the bioequivalence while developing our topical products. I've listed here some of the examples or a screenshot I have for a couple of products. You can see that here sucrose stearate, distearate, PG stearate, PG100 stearate, and glycerol stearate. Some of the RLD label just mentioned the carbomer. So this just for information purpose I have listed here. Now, when it comes to Q2, quantitate some of the excipients, there are also several challenges exist. I have listed some of them here. If you can see that for polymers, they are complex in structure. They have different end groups, branching, functional groups. So it is very difficult to identify or reverse engineer the polymer to quantitate polymers. Same as polymers, there are oleaginous compounds, as I think previously mentioned in Vaibhav slides as well, that petrolatums, mineral oils, waxes, they have they are also difficult to quantitate, specifically when they are used as a single excipient or in combination. Some of the challenges exist with the buffering agent, specifically if they are used in combination, like citric acid, sodium citrate is used. They are interconvertible and in the formulation. So when we reverse engineer those, it is extremely difficult for industry to identify those or quantitate those excipients. Same way, some of the excipients are very low in concentration, like polysorbates, some of the surfactants, some of the carbomers. So in that case, it is difficult for industry to quantitate them. So these are several challenges exist for industry to qualitate and quantitate the innovative products. With that, I would like to move to a case study that I'm going to present here for one of the semi-solid products. Innovator has used particular grade of carbomer. I have just listed grade X and Y for illustration purpose, but Innovator has used the grade of carbomer with the benzene in it and as a polymerization solvent. When we are trying to develop a generic product, due to restrictions on benzene grade carbomer, we have to replace that with ethyl acetate polymerization solvent grade of a carbomer. So the expectation here was that both the grades are interchangeable, and then we have reverse engineered all other components as well, including carbomer with some of the orthogonal techniques that we have available, and had a good success in that. So we reverse engineer all the excipients and try to develop a product. When we manufacture a product and then we achieve the physical and chemical property same as RLD, here is a summary of some of the physical characterization that we perform. If you see on left slide, RLD we have established some range depending on multiple lots available for pH, viscosities, flow curves, and specific gravities. And we have developed a product with reverse engineering of our excipients, including carbomer, and use the same concentration of carbomer, but with the identical grade. So the product we developed also matched the physical chemical properties as RLD, and it was within the well within the range of RLD lots. So with that, we have decided to go into the clinics and perform the pharmacokinetic study. If you can see in this particular slide, and the results are like Cmax and confidence interval both fell outside the acceptance criteria of 80 to 125. There is a high intra-subject variability observed as well. However, the data indicates that the failure was not only due to the lack of sufficient power but also it shows that both the formulations were different and test product were releasing faster. So we have from this data, we have relooked all the information that was available, including reverse engineering, carbomer data, and everything seemed to be well. Then we have decided to develop in-house characterization method, in vitro release method, and see that whether or not do we have a method that can help us to see the difference in the lab as well. So we decided to use a IVRT method in R&D scale, and to see whether or not can we see a similar observation in the lab as well. And we did see that the test product released faster than innovator product. If you can see, the slope was higher for the test product. And we tried to generate some more data apart from IVRT. So we generated some of the drying rate data, and we compared the drying rate of test product and reference product. If you can see in red is the test product and reference product. So there was some difference in the drying rate as well, but we were not, however, we are not sure whether or not it is significant or not. So with all the information, we have decided to perform some of the R&D studies and with different concentration of carbomer and compare the release rate. And based on the some of the R&D experiment, we decided to increase the carbomer concentration, which is outside of those 5% plus or minus 5% level. And with higher concentration of carbomer, if you can see, we performed the IVRT study in R&D scale and see whether or not do we see that difference of a carbomer concentration in the release or not. And we see that difference, that we see RLD slope, and then newly developed reformulated product is closer to the innovator drug. However, previously formulated product was faster. This is one of another set of data where we tried to run a study against the test product of old test product and new product, and we've seen similar difference as well. So we can see the clear difference in old and new formulation, and we have also evaluated some of the physicochemical properties as well. Some of the physical properties I have listed here: previously formulated product and newly formulated product. If you can see, newly formulated product also fell within the within the RLD ranges with slightly higher viscosity because of higher concentration of carbomer, but it was still fell well within the different lots of variability of RLD. Also, we have studied that drying rate here. Now, if we can see that before previously formulated product in orange or yellowish, and then a newly formulated product in purple and reference in green. So the newly formulated product, we see a closer drying rate in comparison with a initial test product. So we can see that it was getting closer compared to the other product. So with this information, with this confidence, we have gone back into the clinics, and then we have performed some of the pharmacokinetic data, and then you can see in some of the data based on this study, SWR of Cmax, AUCT, and I is greater than 0.294. So we scaled average bioequivalence approach was established, and 95% confidence interval is less than or equal to 0. So the study passed statistically. So from this information, we can say that even though the polymer grade was like to like a replacement, it did not work with this product specifically, and we needed to go outside of those 5% level to achieve the bioequivalence. So now I'm going to talk a little bit about, of course, Samir and Vaibhav discussed already on, and Dr. Raney as well, on this particular guidance that just came out 10 days ago from the agency. So recently, FDA came out with the guidance on physicochemical and structural characterization of topical products. And the question is, can we use Q3 similar approach where Q1 and Q2 cannot be established because of the challenges, and then cannot be established within 5% and still show that the test and reference product are same. So in terms of material attributes and its impact on bioequivalence, and also as discussed by Dr. Raney, that how to avoid potential failure modes. For this presentation purpose, I have called some of the excipients which are critical for bioequivalence as a critical excipients, and some of them which are not critical excipients for bioequivalence I have called as a non-critical excipient. So with this thought, I would like to discuss that whether or not is there a possibility where some of the excipients which are not critical in terms of product performance or product bioequivalence, like preservatives, can we have a product which falls outside of those 5% if we avoid those failure modes, like if it's below the IID levels, if it is stable system throughout the stability, if preservative effectiveness test at 50% passed, and with specific product specification, do we have a way to go forward developing a product outside of those 5%? Same way for antioxidants, if we avoid those failure modes, like if it's below IID, if it's stable formulation, and if it's effective throughout the stability at the lowest concentration, can we go and formulate a product outside of those 5%? Buffering agent as well, it can maintain the pH and the buffer capacity throughout the stability. And if it is below IID, do we have a chance to go outside of those 5%? These are some of the excipients that I've discussed, which are less or non-critical to the product release. And when we talk about some of the excipients, as I explained in the case study, that like carbomers and viscosity builders, do we have a pathway that if we can avoid those failure modes, and if we have a way, if we can fall outside those 5% and avoid these failure modes, like if it's below IIDs, if they are Q3 comparable, and if there are available techniques like IVRT, IVPT, the newly developing technologies like DOFM, thermodynamic activities, is there a way we can utilize one or more in combination and approach for a product development? So with this, I would like to summarize today's talk is that there are several excipients that it is difficult to characterize those qualitatively and quantitatively, and in that case, if for some of the non-critical excipients for product bioequivalence, do we have a pathway to develop the product outside Q1, Q2, and whether if they are critical in terms of product bioequivalence, can we use any of the alternative approach, either alone or combination and develop our generic products? With this, I would like to thank Solaris team, Dr. Lakshmi Raghavan, and entire team for helping and putting this together. And thank you very much.
Thank you so much, Mr. Jani. We are now going to switch gears a little bit and move over to some of the research we have been doing in the GADUFA Regulatory Science and Research Program. Our next speaker today is Dr. Narasimha Murthy, who is the Chief Scientific Officer of Topical Products Testing, LLC. Dr. Murthy was a former professor of pharmaceutics and drug delivery at the University of Mississippi School of Pharmacy. He is also the founder director of a nonprofit research organization, the Institute for Drug Delivery and Biomedical Research in Bangalore, India. He has over 150 research papers and publications, and he has also received awards such as the Global Indus Tech Innovator Award from MIT. He's an AAPS fellow. And in 2022, he was recognized with the AAPS Pharmaceutical Global Health Award. So please join me in welcoming Dr. Murthy. We will be hearing a recorded presentation for him. Dana?
Hello everyone, my name is S. Narasimha Murthy. I'm the Chief Scientific Officer of Topical Products Testing LLC, a contract research company located in Oxford, Mississippi. We provide contract services to pharmaceutical companies in terms of developing topical products, characterization, as well as evaluation of topical products as well. So today I would be talking about influence of progressive change in the degree of saturation of API at the site of application on the performance of topical products. This is a typical infinite dose study setup. Basically, in this infinite dose permeation studies, we place adequately high amount of formulation in the donor compartment, and we occlude the donor compartment to mitigate any loss of solvents from the formulation. And then we sample the receiver medium at different time points and measure the amount of drug at different time points to determine the permeation rate and the extent of permeation. So in this kind of a setup, generally the permeation rate and the extent of permeation depends on the concentration of drug in the formulation, because the concentration of drug is proportional to, or it reflects the thermodynamic activity of drug in the formulation. When we are comparing the permeation rate and the extent from two different solvent systems per se, then we have to consider the degree of saturation, because the degree of saturation truly reflects the actual thermodynamic activity of drug in those two solvent systems. Meaning that, degree of saturation is typically defined as the ratio of concentration of drug to the saturation of solubility of drug in that particular solvent. So typically that was an infinite dose permeation study, but in the real life situation, you apply a small dose of product on top of the skin, and the formulation would start losing the solvent, the volatile components in the formulation from the time you apply the formulation onto the skin. And due to the evaporation of solvent, the composition of the product would change eventually, leading to a change in the critical quality attributes. For example, this particular diagram here is representing the entire the changes, the critical quality attribute changes occurring in the formulation at the site of application due to evaporative metamorphosis. So the viscosity of the formulation would change, and the drug distribution between the oil and the aqueous phase, if it's a cream product or a lotion product, could happen. And there could be change in the adhesion of the formulation onto the skin surface. Particle size may increase with time. The globule size and size distribution would change as well. So what is more important for us in this particular presentation, from the perspective of discussing some of the case studies, is the changing thermodynamic activity of the drug, or a degree of saturation of the drug in the formulation at the site of application. So how would the degree of saturation of API change with the time, and how would that change influence the performance of a topical product is a focus in this presentation. So let us start discussing what happens when you do a finite dose permeation study practically. So let us consider a single solvent system. Basically, like, there are only two components. The drug is dissolved in a solvent system, like maybe alcohol or water. So when you place a finite dose of formulation in the donor compartment, the solvent would evaporate with time. And per se, you are starting off at a 50% saturation level of API in the solvent system. The solvent evaporation would lead to decrease in the volume of the formulation. Decrease in the volume of formulation would lead to increase in the drug concentration, isn't it? So basically the increase in drug concentration here means increase in degree of saturation also, because you have the same solvent system all along, and the solubility of drug in the solvent system remains constant. Only the concentration of drug is changing over a period of time. The ratio of concentration to degree of saturation would also change, would also increase with time in this case. So typically the rate of permeation of drug, the transdermal flux, what we call as, is directly proportional to the rate of increase of concentration of drug, or the rate of increase of degree of saturation of API in the solvent system. So this is a diagram that represents the finite dose permeation studies. when you have two solvent system, a binary solvent system, right? So we have two solvent systems, solvent A, solvent B. I considered solvent A as a low boiling liquid and solvent B as a high boiling liquid. So obviously would expect when you apply this formulation, at finite dose, you would expect the solvent A to escape relatively faster compared to solvent B, right? So in this picture, you see that, in the circles, you start off with like 25% mole fraction, or 0.25 mole fraction of solvent B, and the mole fraction would go on increasing with time. At T1 and T2, you see that the mole fraction of the solvent B is getting more and more. So you also observe that the size of the circle is decreasing, representing the volume of the formulation is decreasing as well. So we know that from the previous slide that as the volume decreases, the drug concentration would increase. If the solvent B is acting as a co-solvent in this formulation per se, then the increase in the mole fraction of solvent B would lead to a significant increase in the saturation solubility of drug in the remnant formulation at different time points. So what would happen due to this? Although there is an increase in the drug concentration because of the increase in the drug solubility in the remnant formulation, the ratio, the degree of saturation would go on decreasing with time, right? You start off with a certain degree of saturation, but because of the escape of the low boiling liquid and increase in the co-solvent concentration, the solubility of the drug is rapidly increasing. This would lead to decreasing degree of saturation with time. And although we see an increase in the drug concentration, the degree of saturation decreases, and that could negatively impact the rate of permeation of the API from such formulation. So to demonstrate this phenomenon of evaporative metamorphosis from a binary solvent system, we considered two solvents here: water low boiling as a low boiling solvent, and PG200 as a high boiling solvent. And then we designed the experiment in such a way that we would be able to sample the formulation from the donor compartment to measure the water concentration, the PG200 concentration, as well as the drug concentration in the formulation at different time points, in the formulation in the donor compartment at different time points. Of course, we also measured the drug concentration in the receiver medium to eventually be able to draw the flux time profile. So we had to take kind of little higher amount of formulation than the typical finite dose definition describes. If you take like 15 milligrams per centimeter square, you would not be able to sample the donor, the formulation from the donor compartment at different time points and determine the concentration of all these different components. So we had to take about 300 micrograms of formulation in the 300 micrograms per centimeter square of formulation in the donor compartment, and left the donor compartment open, unoccluded, so that the solvent would evaporate and we would be able to determine the drug concentration and the solvent composition at different time points to be able to calculate the degree of saturation of drug at different time points. As a part of this experiment, we also determined the solubility of metronidazole at different concentrations of PEG in water. The graph on the left shows you how the solubility of metronidazole changed as we changed the PEG concentration in water. So I'm considering two representative formulations. Although we prepared multiple formulations, I'm just considering two formulations for ease of explanation. So we have one that is like 30% PEG formulation and a 40% PG formulation. In both of them, we had about 70% of metronidazole or metronidazole concentration at 70% of its saturation solubility. So you see the way the degree of saturation on the right side graph, you see the way the degree of saturation changed over a period of time. So we sampled the donor compartment at different time points and measured the PEG concentration, the water concentration, as well as the API concentrations. And we know the concentration of API at different time points, and we know the percentage of PEG remaining in the donor compartment, in the formulation in the donor compartment at different time points. So using the composition of the formulation, the solvent composition in the donor compartment, and using the concentration of drug at different time points, we would be able to calculate the degree of saturation and draw this, plot this graph of the time course of degree of saturation of metronidazole in the formulation. So the point I'm trying to bring up here is, although there is only 10% difference in the PEG concentration, when you start off, eventually, due to the different rates of drying, due to the different rates of change of concentration of drug and the rate of change of solubility, the degree of saturation profile is significantly different between these two formulations. So just to give you a little bit more clear idea, I've put together a table here to explain how we calculated the degree of saturation profile. So the first column is the time points, and the second column is the percent PEG concentration, the change in percent PEG concentration in the donor compartment. And once we know the percent PEG concentration at different time points, you will be able to determine the solubility of drug in the PEG solution using the solubility curve. And then we know the drug concentration because we determine the drug concentration practically in the formulation that is sampled from the donor compartment. So you know the drug concentration, you know the solubility of drug, and the ratio would be degree of saturation. You see how the degree of saturation goes on changing in case of 30% formulation and 40% formulation. So although we start off with the same degree of saturation of 0.7, eventually, the way the degree of saturation is increasing in case of 30% formulation is way higher, significantly higher compared to the 40% formulation. So here is the transdermal permeation flux of API, the metronidazole from two different formulations. The time course of flux profile of these two formulations was significantly different, and it was very much coinciding or corresponding to the time course of degree of saturation of API that we observed in these two formulations, as I shown in the previous slide. So basically, the difference in the performance between a 30% PEG solution and a 40% PEG solution of metronidazole is attributable to the time course of degree of saturation, the way the degree of saturation changed over a period of time, right? But there is one caveat here. So here on the right side graph, you see the viscosity of different concentrations of PEG 200 in water, right? So you see that the viscosity of PEG 200 in water goes on increasing as you increase the concentration of PEG 200. So in our experiment, you see that the PEG 200 concentration would increase with time as well, right? Due to escape of water, the mole fraction of PEG 200 would go on increasing. So obviously, the formulation viscosity also would be increasing with time in the donor compartment, besides the change in the degree of saturation, right? So there are two variables occurring here. One is the degree of saturation changing with time. The other one is changing viscosity in the formulation. So the difference in the performance between 30% formulation and 40% formulation cannot be entirely attributed to the time course of degree of saturation alone, because there is one more variable, the viscosity changes, that we have to either consider or account for, or we have to completely rule out. To investigate the role of changing viscosity or the influence of changing viscosity on the permeation profile of metronidazole from 30% and 40% formulation, we did further experiments. more like a mechanistic studies to understand the role of viscosity on the permeation of metronidazole from the formulation. So to understand the role of viscosity on the permeation profile of metronidazole from PEG solutions of different concentrations of PEG 200. So we prepared PEG 200 solutions with different mole fractions in water, and then we incorporated metronidazole at a degree of saturation of 0.5. And if you observe the viscosity of the formulations was significantly different across all these formulations from 15% PEG to 85% PEG. And then we performed IVPT study using infinite dose of formulation, so meaning that we had 500 milligram of formulation per square centimeter in the donor compartment, and we had occluded the donor compartment to mitigate any solvent evaporation. Then we looked at the amount of drug permeating across, over a period of time, like we do, in case of conventional infinite dose IVPT study. And then we found that there was no significant difference in the permeation profile of metronidazole from different PEG 200 solutions. So what does this tell? This tells us that the viscosity had no role in determining the permeation of metronidazole from different concentration of PEG 200 solution. Meaning that in the previous experiment where I showed you 30% and 40% formulations, the IVPT profile of 30% and 40% formulations at finite dose, you see the viscosity would increase due to increase in the mole fraction of PEG 200, but would not influence the permeation of metronidazole, and the difference in performance the difference in performance between 30% and 40% formulation that we observed in the finite dose IVPT study is entirely attributable to the time course of degree of saturation, and viscosity has no interference or no role in determining the permeation of metronidazole from this 30% and 40% formulation. Hope it is clear. So after looking at the solution systems, we upgraded to gel systems. Typically the same solvent system of PEG 200 and water. We incorporated a gelling agent, the hydroxyethyl cellulose, which is a good hydrogel agent. And we also incorporated a couple of more ingredients, EDTA and sodium benzoate, just to preserve the formulation. And this time we considered 3 APIs for the study. Metronidazole again, and we considered diclofenac sodium, and then lastly we also considered lidocaine, the base, not the salt of lidocaine. And the bottom you see 3 tables corresponding to 3 different APIs, and showing the characteristics of these formulations. So let's look at the metronidazole table. We incorporated different amounts of PEG in these formulations, 110 and 20%. Metronidazole concentration was same across all these formulations. The degree of saturation was calculated based on the solubility of drug in the formulation. And typically, the reason why we selected the PEG concentration to be between 1 and 20% was to have formulations with same Q1 and significantly different Q2 and a comparable Q3. If you look at the viscosity and the pH values, they did not really differ significantly across all these 3 formulations. So we had 3 different formulations prepared with different concentrations of PEG 200. Otherwise, the rest of the composition is the same amount of drug, and all of them had comparable quality attributes. So before we subjected the gel formulations to finite dose IVPT, we performed some infinite dose IVPT studies using these gels. We considered 3 different degrees of saturation for each of these gels, meaning that we incorporated the drug at different levels in the gel system. The entire composition was same, just the amount of drug that we incorporated was different to create a gel system with different degrees of saturation. And then we determined the steady state flux. So because this is an infinite dose IVPT, you see that the flux would become constant after a period of time, and then it would kind of remain consistent over a long period of time. We averaged the steady state flux and then we correlated the average flux, the steady state flux with the degree of saturation of API in the formulation. So we found that in case of all these three drugs, the metronidazole, diclofenac, and lidocaine, we found that there was a good correlation between the transdermal flux of the API and the degree of saturation of API in the formulation. So this is a basic requirement for the drug to be a good candidate to evaluate our hypothesis in this experiment. Okay, so meaning that all these drugs, the transdermal permeation of all these drugs is quite sensitive to any change in their degree of saturation in the formulation. So that's a basic requirement for us to go ahead with the further experimentation. So that factor is ascertained here. So that fact is ascertained here, and then we perform the finite dose IVPT studies after assessing the suitability of drug by performing infinite dose IVPT studies. So let us now look at what happened in case of finite dose IVPT studies, case by case basis. So let's take up the metronidazole gel first. So what you see here is the change in the concentration of water on the left side graph, and the change in the concentration of PEG at different time points on the right side graph. So as I showed in the table, we had 3 gels prepared for each of the API. So the 3 gels contained 1%, 10%, and 20% amount of PEG 200 in them. The amount of API was same across all 3 gels. So in the left side graph, you see the concentration of water is decreasing with time in case of all 3 gels, and likewise on the right side you see the PEG concentration is increasing with time in case of all these 3 gel systems. So the water decreasing, and that's why the PEG concentration is increasing. Okay, so you remember that we also measured the drug concentration in the donor compartment, in the formulation in the donor compartment at different time points. So now we have the concentration of drug at different time points, and we also have the solvent composition at the corresponding time points, right? So once we know the solvent composition from the right side graph, the solubility graph, which I had shown you previously also, we will be able to calculate the amount of the saturation solubility of API in those solvent compositions, the remnant solvent compositions at different time points. So you know the drug concentration, you can calculate or you can determine the solubility of drug from the solubility curve, and then you can calculate the degree of saturation and plot a time course graph of degree of saturation. So let us see what happened in case of each of these formulations. So 1, 10, and 20%, obviously you see the degree of saturation went on increasing with time, but there was no significant difference in the profile of degree of saturation across these formulations. All the profiles, the degree of saturation time profiles of 1%, 10%, and 20% were intervened, and they were overlapping. So what this tells us is that the degree of saturation of API in these formulations, despite having different amounts of PEG 200, did not differ significantly, and it is less likely that these formulations would perform differently when you do the finite dose IVPT studies. So in this, you see the permeation flux profile of metronidazole from gels containing different amounts of PEG 200. So from the graph as well as from the dermatokinetic parameters that are tabulated on the right, you see that there was no significant difference in performance between these formulations. So they all perform pretty much the same way. So this is this was predicted based on the time course of degree of saturation from the previous slide. I showed you how the time course of degree of saturation were overlapping in all these 3 formulations, in case of all these 3 formulations, and we kind of predicted that these 3 formulations are less likely to perform significantly different in terms of bioavailability, in terms of the permeation flux time profile. That is what exactly was observed, right? So the important observation we made, or the important lesson we learned from this experiment, is that in all this in all these three formulations, the composition pretty much remained the same, because the amount of drug is same on all the Q1 is same. Only the Q2 was different, significantly different, right? The PEG 200 concentration, we varied from 1% through 20%. Despite the difference in the concentration of one of the ingredients, all these three formulations resulted in almost overlapping time course of degree of saturation. So when you have such an overlapping degree of saturation profile across formulations, despite having a difference in the concentration of one of the ingredients, they are less likely to result in significantly different bioavailability, meaning that their bioavailabilities would be somewhat comparable, like we saw in this case, right? Now let us look at what happened with the diclofenac gels. So typically same type of formulations, 1% to 10% and 20% PEG with the same amount of API. Rest all the ingredients were at the same levels across all these three formulations. And again, like we saw in case of metronidazole, we observed a decrease in the concentration of water and increase in the concentration of PEG with time in the donor compartment formulation. So this is the degree of saturation profile of diclofenac sodium in case of different gel formulations. So here you see in case of 10% and 20% PEG containing gel systems, there was no increase in the degree of saturation. But in case of 1%, there was a progressively there's a progressive increase in the degree of saturation with time. So why did this happen? Let's look at the solubility curve to understand why there was no increase in the degree of saturation in case of 10% and 20% gel, and why did we see progressively increasing degree of saturation in case of 1%. So in the solubility curve on the right side, you see the solubility of diclofenac sodium at 32 degree at different concentration of PEG 200. I kind of zoomed in the solubility curve on the top graph just to consider the PEG concentrations up to 30%. So let us take a look at what happened at 10% and 20%. So in case of 10% formulation, you see that the concentration of PEG 200 increased from 10% to maybe 15% or 17%. So due to this increase in the PEG concentration, the drug solubility increased significantly. Same thing happened with 20% also. From 20%, the concentration of PEG increased, the mole fraction of PEG increased due to evaporation of solvent. So this increase in the PEG concentration led to a drastic increase in the solubility of the API. So, but in case of 1%, the solubility did not increase significantly because the 1% of PEG solution only increased from 1% to 2 or 3 or 5%. So across this concentration range of PEG, the solubility remained pretty much consistent. So although there is an increase in the concentration of drug due to evaporation of the solvent, because the solubility remained pretty constant in the donor compartment in case of 1% formulation, the degree of saturation was found to increase with time. But that didn't happen with 10% and 20% because relative to the increase in the concentration, the solubility, the increase in solubility was relatively more drastic. So that's why the degree of saturation was either consistent or showed a kind of a downward trend in case of 10% and 20% gel formulations. So now you look at the bioavailability profile of diclofenac sodium from different gels. You see that 1% formulation outperformed the 10 and 20% gel formulations. So the area under the curve, if you look at the 1% had almost 2 times the area under the curve compared to 10 and 20%. So what this tells us is, despite the co-solvent level being less in case of 1% PEG gel compared to 20% and 10% and 20% PEG gel, the 1% PEG gel outperformed the other two, mainly because in case of 1% PEG gel, the evaporative metamorphosis led to a drastic increase in the degree of saturation, leading to increasing thermodynamic activity, which in turn resulted in a much better performance in case of 1% gel compared to the other gels. We observed a similar phenomenon in case of lidocaine gels as well. In case of 10 and 20% PEG containing gels, we did not see a significant increase in the DOS, the degree of saturation with time. But in case of 1%, we saw a drastic increase. At fourth hour, we saw the degree of saturation attaining a plateau, meaning that the degree of saturation was 1, and beyond that point, it would either remain the same or it would become supersaturated if the solvent is available, until the point that the solvent is completely depleted in the formulation and the drug would precipitate out of the formulation. So why did this happen? Let us look at the solubility curve of lidocaine at different PEG concentrations, the PEG 200 concentration. Please take a look at the zoomed in graph on the top. So what this tells us is, in case of 10 and 20% PEG solution, any increase in the PEG concentration due to evaporation of water led to a drastic increase in the solubility. So therefore, the degree of saturation in case of 10 and 20% gel formulations dependent predominantly on the solubility of drug in the remnant formulation at different time points. But in case of 1%, because the solubility from 1 through 5% remained almost consistent, so the 1% PEG would have increased to 2 or 3%, did not lead to significant increase in the solubility of the drug. So therefore, the degree of saturation in case of 1% PEG containing gel formulation predominantly dependent on the concentration of drug in the formulation at different time points. So even in case of lidocaine gels, similar to diclofenac sodium gels, we saw 1% PEG containing gel outperformed the 10 and 20% gel. Now you know the reason. In case of 1%, we saw a drastic increase in the degree of saturation with time as compared to 10 and 20%. So that's why the area under the curve in case of 1% gel was 2 times that of 10 and 20% gels. So this is another case study which proves our hypothesis that the time course of degree of saturation is one of the most important or critical factor that would determine the performance of the formulation, despite there is a difference in the composition of the formulation. If the formulations result in comparable time course of degree of saturation, their bioavailability would also be comparable. If there is a significant difference in case of time course of degree of saturation across these formulations, it would end up resulting in a significantly different bioavailability profile. So I would like to conclude by saying that in case of drugs such as metronidazole, lidocaine, and diclofenac sodium, when the formulations have comparable critical quality attributes, it is the time course of degree of saturation that determines the rate and extent of drug permeation from the formulations. And if you've observed that all the formulations that we explored so far had the same Q1, same almost matching critical quality attributes, only Q2 was deliberately varied by varying the concentration of PEG 200 in such formulations. Investigating the degree of saturation time profile of products would enable us to predict if the products would end up being bioequivalent or not. So to assess our hypothesis further, to add more supportive evidence to our work, we are going to work, with the other solvent systems, propylene glycol and PG400 and other solvent systems. And maybe we would also add a couple of more APIs to our experimental design to generate additional data to support our hypothesis. I would like to acknowledge the grant support from US FDA, the 2 UO1 fundings, and I would like to thank the TPT team and the wonderful people on the FDA team as well. Thank you so much.
Thank you so much, Dr. Murthy. We are running a little bit behind, but we will be using a little bit of the lunch break to continue with our panel session. But before that, our last presentation for this session today, our speaker is Dr. Yousuf Mohammed. Dr. Mohammed is a research leader at the University of Queensland. He currently leads multiple FDA-funded research projects to enhance or impact regulatory guidelines for topical and transdermal products. Over the last decade, his work has focused on dermatological product development, assessment of performance, drug-device combination products, skin permeation assessments, and assessment of toxicology of drugs. Dr. Mohammed completed his PhD in pharmaceutics and skin drug delivery in 2013. Please join me in welcoming Dr. Mohammed. Should we go ahead?
I'm sorry, everybody. I think I was muted. Thank you so much, Priyanka. Good morning, good afternoon, and good evening, everyone, wherever you're listening from. And if you're anywhere close to where I am, I'm grateful that you stayed up to listen to these excellent speakers. I'm Yousuf Mohammed from the University of Queensland in Australia, and we partner with the US FDA to deliver high-quality research that enables recommendations to be made. Today, I'm going to be talking about development of methods for evaluation of formulation differences and their impact on therapeutic equivalence, and in particular about how we could broaden the therapeutic scope. All through the morning session, you've been listening to high-level talks from eloquent speakers who've been discussing the implications of formulation on product performance. We've also heard about the Q3 guidance and what it means. And if we come to the Q3 guidance, the primary question over there is the arrangement of matter the same? And when this question is asked, we very quickly come to an understanding that the arrangement of matter is to be assessed at multiple levels. It can't be done at a single level. When it comes to product quality and performance, the product quality and performance are closely related. In fact, the new Q3 guidances recommend Q3 sameness and emphasize on physical and structural similarity. If we were to retrace our steps to the regulatory recommendations, sensitive and discriminatory Q3 tests are at the heart of the in vitro approach. Hence, developing a battery of tests characterizing the physical, structural, and microstructural attributes are imperative. In fact, sameness within the range characterized for the reference standard has the potential to guarantee therapeutic equivalence. Talking about broadening the therapeutic scope, even before a topical product, in this case, we could say a generic product, is applied to the skin and owing to its skin penetration, exerts its therapeutic effect, it first comes in contact with the patient through sensory means. And the new age consumer or the patient sees beyond the bitter pill logic. Traditionally, medications were used as bitter pills, which we had to consume to get better. But is that still true? I wouldn't think so, because topical medications are no exception. If a pill requires masking of taste through excipients for compliance, so do topical formulations. In particular, generic products, therefore, would require to look and feel as elegant as a reference product, at least. Patient skin can often be irritated and damaged, and hence a cooling product can start to provide its therapeutic effect long before the drug penetrates the skin. Similarly, the perception of grit or grittiness can further irritate sensitive skin and sore skin, and therefore be therapeutically a negative experience. And this will induce a nocebo effect, which would reduce compliance at the next level. So the question is, can we design Q3 tests that cover a larger area of therapeutic equivalence? Firstly, what are the different sensory attributes that are perceived? At the top of this list is perceivability itself. When you put a semi-solid product onto the side of application, do you even feel it? The next thing is, what does it feel like? A range of perceptions are evoked. For example, hardness that can be tested through texture analysis is the force exerted to apply the product. And cooling sensation can arise due to temperature difference between the product that is often applied from room temperature onto our body, which is at a higher temperature. Also, a volatile any volatile ingredient of the formulation as it evaporates, this cooling sensation is enhanced. In fact, there is grittiness, absorption point, velvety soft feeling, so on and so forth. We'll probably end up covering most of these today, if not all of them. Now, if we look at the mechanism behind the effect itself, this will give us a good indication of what instruments can possibly be used and what the results can tell us. For example, stickiness of a product can be analogous to the initial viscosity or the zero shear viscosity, which is a standard rheological test, and it's a little bit more informative than apparent viscosity that can be measured using a rotational viscometer. Another example is quick drying of the product. Now, when it comes to quick drying of the product, it's a sensory perception. At the same time, in terms of Q3, we know that drying rate, just a simple drying rate test that Dr. Murthy talked about, or the previous speakers have been talking about, can give us a lot of information. Analyzing this also has important clues on how therapeutic effects or therapeutic equivalence can be impacted in generic if these attributes are not matched. Therefore, the sensory attributes are not just sensory, they have a lot of therapeutic implications as well. We'll look at some of these in detail. So one of the reasons for grittiness in a product is inconsistencies, and this can often mean presence of crystals or large lumps. Over here, the crystal size, aspect ratio, and the crystal habit definitely have an effect on therapeutic equivalence. Grittiness can also increase irritancy when a product is used on an already irritated skin. The other spectrum of grittiness is softness. It is already known that products that feel soft can hydrate the skin, and it is known that skin hydration enhances penetration. Especially if you have a thick layer of product, skin hydration via occlusion can also temporarily alter the skin barrier properties and allow for an enhanced flux, and this is true for both hydrophilic and hydrophobic compounds. In fact, the emollients and surfactants used to improve the skin feel can have penetration enhancing effects. Let's talk about cooling sensation. Cooling sensation, it can be due to the alcohol content, and this can lead to differences in solubility if you look at therapeutic properties. Cooling can also reduce itchiness and irritation and complement the efficacy of API in certain skin conditions where surface evaporation or cooling has got a soothing effect, and this is positive. So not all sensory attributes have got only positives about them. An individual sensory attribute can have both positive and negative impacts. Let's take greasiness, for example. In most cases, this would mean that the greasiness is first off starting off as a product of high viscosity of the product. Increased viscosity, we all know, can lead to reduced release from the formulation. However, enhanced occlusivity due to this product can then improve penetration. In fact, I was just listening to the chats and somebody was talking about a high viscosity product. I think it was Dr. Guy. And one of the good positive side effects of this is that there'll be less wash off effect, and the product will almost act like a water resistant product, and it will lead to a higher wear time on the skin ultimately affecting the therapy. So all morning, like I said, we've been talking about Q3 tests, and I would like to call the test that I'm going to describe in the next few minutes as Q3 plus, because we've made some modifications and enhancements in the procedure, and this definitely, this definitely gives us additional capabilities and allows us to capture a fuller picture. So one of the first tests that we will talk about, let's start off with one of the most common recommendations in Q3, measurement of volatile loss. This is typically done on a range of formulations, which may or may not be Q1 and Q2 the same. Now we need to understand that the work needs to be done in replicates, which is standard.
Quiet.
For the test itself, a known amount of product is placed on a pre-weighed glass slide, and a gravimetric loss is assessed over time at one or more temperatures. As an additional setup, we could do it where we spread the product in a thin layer using a micrometer applicator. So now even the thickness is a known commodity, and the evaporation is assessed in more in-use-like conditions. The next modification we can do is that to the first setup, if we added a couple of thermocouples, one at the top and one at the bottom, we can assess temperature differences in the evaporating top layer, as well as the bottom layer that is although protected, but it's in contact with skin, and the temperature of that will increase much faster. You all know what a typical setup for a texture analyzer looks like. A standard test that we do would be a compression test. And in a compression test, firmness is the maximum force required for the compression to set. Spreadability is the magnitude of shear work required for the first cycle of compression, and it is denoted by this area, A1. Adhesiveness is the magnitude of work required to withdraw the probe from the original height after the first compression, and area A2 signifies this. Stringiness or tailing is the distance to which the product remains adhered to the probe during withdrawal after the first compression, and that's signified by this distance D. We know that these textural properties can be linked to various aspects of the formulations. Among other things, we looked at polymer content of the gels and the strength and appearance of the polymer network. We developed sensitive techniques to study these polymers using cryo-SEM and employed a range of freezing, fracturing, and sublimation techniques under a range of conditions. Images at the same magnification that you're seeing over here show how polymer content leads to high and low pore density. And obviously, therapeutic implications of these can be very well understood. The next range of Q3 tests are rheological assessments. A full rheological profile, including oscillatory strain sweeps, temperature sweeps, rotational thixotrophy, shear sweeps, are standard tests that we routinely conduct. So today, instead of going into the methodology, which is detailed very well in literature, as well as it's available on various other platforms, I'll dive into the implications and connections to sensory perceptions, as well as therapeutic considerations. So viscosity has a positive relationship with sensory tackiness and stickiness, and it has a negative relationship or an inverse relationship with spreadability and oiliness. Other parameters that we regularly assess are storage and loss modulus. So all of you would appreciate that if the G prime is greater than G double prime, this shows that the sample is a viscoelastic solid, where the tan delta is less than 1. Whereas if the G double prime is greater than G prime, sample is more viscous than elastic. We all know the importance of the viscoelastic region. Zero shear viscosity is another thing. The relationship over here is the zero shear viscosity is related to the first pickup of the product. That is when the user first encounters the product and its inherent strength. On the other side of the spectrum, infinite shear viscosity is a measure of friction and is related to in-use product behavior while rubbing the product onto the skin just before it disappears. We all know that yield stress is a good measure to understand spreadability and ease of application. And as it can be appreciated, temperature sweeps are done to understand the changes in critical rheological parameters between a specific temperature range to try and simulate temperatures that can be achieved while the product is being used, especially when the product is being rubbed and a little bit of heat is exerted. Now you all know and appreciate rheology, but another sister of it that doesn't get appreciated as much is tribology. It's a less common yet very important assessment of flow sweeps to assess lubrication provided by the formulation. We've developed a range of methods on different instruments and use biological and non-biological material as bottom surfaces to try and study the relevance to lubrication on the skin itself. A typical strieback curve that we're looking at over here has three distinct regions. A boundary area where there is material, in this case, we're talking about formulation in between the finger and the site at which the formulation is being applied. Now, if this material provides lubrication, friction goes down very quickly. Once the material is spread and viscosity is dissipated, friction very soon starts to go back up. And this depends on the nature of the material. Creams, lotions, ointments, which have got oleaginous substances, often form a film between the two surfaces, and these tend to lubricate for much longer. Gels, on the other hand, as you appreciate, dry out quite fast, and you see a very steep rise in friction in gels. And this is just like what we see in vivo. Like I mentioned, we've utilized a long list of conditions, materials, and instruments to simulate skin-on-skin kind of interactions. And currently we're working on materials that can replicate moisture content, oiliness, and friction based on large number of data that we've generated from large number of volunteers and patients in disease conditions. So eventually we'll be able to tell what a particular skin type will perceive when a product XYZ is applied to the skin. Moving on to this from rheology, tribology onto the big guy, and in the context of this presentation, it's the elephant in the room. Cooling sensation and how we measure it in vitro and in vivo. It's a tricky test to perfect. And for this, we've used a range of thermal cameras, and we started off with mobile phone operated, really low tech devices, and went all the way up to the FLIR T840, which is the state of the art in the market currently. So a typical setup, we first develop instrumental techniques to do in vitro and ex vivo protocol development. And this is often done in parallel to our volatile loss experiments. We have a heat pad, you put a formulation onto a glass slide and look at the temperature differences. Thermal images are captured at any point. I mean, you could do it at 32 degrees, you could do it at 24 degrees and look at room temperature and try and correlate the evaporation data with the thermal imaging data. Once all the big data from all of these Q3 attributes is generated, we need sensitive tools to try and understand and correlate the data to formulations and to use the information to draw meaningful conclusions, as well as select products that can then be taken into the validation step. We first do sophisticated statistical analysis and look at correlation between attributes from different analyses. Similarities between formulations and the role of Q1 and Q2 differences or other components are then assessed. We also look at rank ordering formulations in specific parameters signifying respective sensory attributes. We then look at temporal domain when this attribute will start to appear, because something like cooling sensation and firmness, adhesiveness you can start feeling it from the time you're handling the product. Spreadability, slipperiness, grittiness, evaporative cooling, these things are assessed while you're applying the product. And then rate of absorption, drying, residual stickiness, or residual greasiness, film formation. These are residual properties, and these are assessed later. So if we design test in such a way that these are mixed up, then temporally you don't really understand the impact of each of these sensory attributes, and you kind of miss the therapeutic picture quite soon. So we do sophisticated statistics such as Pearson correlation between material attributes, which then inform us how closely, first of all, each attribute is related to the other attribute. Linear relationship and level of significance are analyzed using Minitab software. The green bits highlight where correlation coefficients are above 9. That means that there is very strong correlation between the parameters. Positive and negative coefficients indicate positive correlation of data or negative correlation with each other. And the level of significance is indicated by stars. And these are all done at 95% confidence interval, where we consider P of less than 0.05 to be significant. And these are the different significance levels that we look at. We then go on and do multivariate clustering of observations using Minitab software package. And in particular, we do centroid linkage and Euclidean distances as analysis preferences. And this gives us unique clusters of formulations that are more like each other than they are different. So this is an example of a zero shear viscosity Euclidean cluster. You'll see that we managed to form 6 clusters. Now we would expect the formulations within the cluster to behave similar to each other. And this helps us a lot in understanding about the formulations, as well as which formulations to take into account. This is an example of similar clustering done on yield stress. And again, this is an example of clustering done on coefficient of friction that is derived from tribology. In the interest of time today, I've not shown all the analyses, but we do, however, do an overall clustering in which all of the parameters, Q3 attributes that we look at are considered. I'll show this in a couple of minutes when I'm talking about the clinical work. So among other analysis, we also do PCA. And for PCA, we consider all attributes, textural, rheological, tribological, cooling sensation, and other Q3 attributes that we look at, pH and everything included. And as you can appreciate, there are multiple tests in each instrument. Rheology itself has 7 different test categories. And you will all know that texture analysis gives multiple parameters. So we really are talking about big data. So powered by this big data, which we then simplified using analysis and statistics, we look at correlation between instrumental analyses and what the actual volunteer or the patient perceives. How this is done, so our overall approach includes manufacturing formulations with careful Q1, Q2, and Q3 variants. And we try and use DOE-like approach to manufacture these. These formulations are then analyzed through multitudes of instrumental analyses. The data is analyzed through sophisticated statistical tests, as well as predictive modeling. And then this information and a curtailed list of products are then taken into the clinical trials. We then understand the behavior and temporal dominance and come up with a qualitative descriptive analysis for the sensory perceptions, where we are able to describe, one, what is the perceivability, what the consumer or volunteer or patient in this case is actually perceiving, and we can actually qualitatively encompass all of these. We then follow this up with sensory panel analysis. And once we're done with that, we trace our way back towards product attributes and make further correlations in vitro, in vivo kind. On the way, we also bring about a huge gentrification of the data by trying to correlate the results that we've got and stratifying the in vivo results according to skin types. We can measure skin hydration, friction, dryness, elasticity on live volunteers, and this data is then used to stratify our results and look at individualized connections. I thought that the best way to cap my presentation today is to demonstrate a case study of gel cooling sensation, where we did sensory evaluation in vivo volunteers. These are the results of the overall clustering of all attributes. And using this, we identified 2 formulations that are highlighted in red in these boxes. And we identified these formulations because these are far enough to each other. We then set up a in vivo volunteer study to see how well our in vivo and instrumental tests are correlated. In fact, with cooling sensation, one of the reasons why we started off with this is there is a possibility to use infrared thermal cameras in vivo to assess both perceptions of the volunteers and actual temperature differences simultaneously. So to do the trial itself, a known amount of product is dosed onto the skin, and after 15 seconds, it's spread over the stipulated area, which is demarcated on the arm. In this case, we used a 2+1 product approach in a double-blinded manner. Parallelly, infrared thermal camera images were captured every 15 seconds for the whole 2-minute duration. The volunteer also fills out a form, which is a sensory evaluation form. And we use a 10-point scale, where 0 being not cold at all and 9 being very cold. Taking a look at the sensory test, the product that was repeated, which is hydroxycellulose gel, compared very well to each other. And we tried to then correlate the 0 shear viscosity to between the difference between 0 shear viscosity in the 2 products and see, because in our in vitro studies using a thermal camera, we observed that higher viscosity products are remaining cooler for longer. So can the volunteer perceive it? And it turns out they actually can. The other sensory parameter that we looked at was evaporative cooling. And the connection of Q2 with evaporative cooling was one of the gels that we did had 50% ethanol, the other gel had no ethanol at all. And you can see that an evaporating product is perceived as cooler. So there is a little bit of that evaporative cooling going on as well. And this evaporative cooling is then paralleled with our water loss or weight loss, volatile loss that we saw, which was largely different between the 2 formulations. Now the interesting part. When we compare sensory perceptions to actual temperature differences between the product that is applied on the application side and the non-dose side, what do we find? Firstly, I'll share some images. These are images from 1 volunteer for the 3 formulations, 2+1. We see that the non-dose spot temperature was consistent within each volunteer forearm. Upon application, the product felt cold, which is an in vivo perception, and a difference in temperature can be seen the initial stages. However, just like in the perception test at 2 minutes, no additional temperature differences can be observed with the infrared thermal camera as well. So indeed, we are able to correlate in vitro and in vivo sensations, where the aim would be in the future to not have to go for clinical studies to do sensory assessments. These can all be done through in vitro measurements. I'd like to say that this is only the beginning and we've got a long way to go. And I'd also like to thank all the people from my lab who helped making this possible. And of course, this work would not have been started or carried out without the collaborative nature of the FDA team or the funding that has given us the resources to carry this work out. Thank you so much.
Thank you so much, Dr. Mohammed. We will now transition into the panel. So if I can please ask the panelists to turn on their videos. And apart from the speakers we had this morning, joining us on the panel today is Dr. Liang. Dr. Liang is a branch chief within the Division of Liquid-Based Drug Products 2, Office of Life Cycle Drug Products, Office of Pharmaceutical Quality at the FDA. So he brings us the OPQ perspective on all of the Q3 discussions you have heard this morning. Now we are running a little bit behind on time, and I'm very, very grateful to all our speakers for continuing to address a lot of questions we received in the chat. So I'm going to try to cover the questions that are overarching and were not covered in the chat so far. So we'll start with a question for Dr. Raney and Dr. Liu, perhaps. We saw some questions in the chat about what do we really mean by arrangement of matter, and specifically, how do we define sameness and similarity? So maybe we'll start with Dr. Raney.
Thank you, Dr. Ghosh. So when we talk about the arrangement of matter, meaning the Q3 attributes, the physical, chemical, and structural arrangement of matter, when we talk about that being the same, what we're referring to is being matched within the ranges that are characterized within the reference product. So for example, if the pH of multiple batches of the reference product is, let's say, pH 7.2, pH 7.2, pH 7.3, well, that defines a range of pH 7.2 to 7.3. And so if the test product has a pH in that range, then it's considered to be the same. And likewise for a lot of the other, particle size distribution and other Q3 attributes. Now, one of the additional things that you might think about, and I would encourage everyone to read the Q3 guidance about the details relating to this, is that while that's the most straightforward way to think about matching the product, it may also be acceptable to be within a range that you haven't been able to characterize as existing within the batches of the reference product that you assessed, but that it would be actually acceptable to FDA for the reference product to be in that range. And this is just as an example, hypothetically, let's say that the reference product would be deemed acceptable at a pH of not only 7.2 and 7.3, but maybe also 7.4. And so in this way, if it was also acceptable for the reference product, it may be acceptable for the test product as well. And this is really when we talk about when the Q3 attributes are matched, and there are a whole series of tests that define these physical, chemical, and structural attributes that define that arrangement of matter. When all of those are matched in this way, then it's considered Q3 the same or similar, depending upon what the underlying matter is, but depending upon the Q1 and Q2. But if those are outside the range that you've characterized for the reference product and outside the range that would be considered acceptable for the reference product, then it would be Q3 different. And that's regardless of what, whether it was Q1, Q2 the same or different. If the Q3 attributes are not all lined up, then it's considered a Q3 different product. So
Sam and Priyanka, I just wanted to harp a little bit about the origin of the term arrangement of matter. So arrangement of matter goes hand in hand with conservation of matter. It goes into the discussion of you pouring a bunch of product, a bunch of excipients and API into a mixer, and you're mixing it or you're manufacturing it in various orders, et cetera. And your end product you have what comes out of the mixer and what ingredients are conserved in the same rate, same quantity. So when you're doing reverse engineering, are they the same amount that you put into it? And then that's conservation matter. And then arrangement matter is, are you getting out of that mixer something that is the same consistency, same look and feel, et cetera, as the reference product. And this goes to the historical comparisons that we used to talk about, and I think we still do, about, for example, cake mixes or cookie mixes. When you're given 2 boxes that are essentially the same, but you're cooking them slightly differently, are you going to end up with a consistent product at the end? And I guess you also have the question of, across different cake mixes, can you end up, in this case, our scenario here that we're discussing today, if you have different cake mixes with slightly different formulations, can you still come up with a cake that tastes and looks and feels and is acceptable for consumption, whether it be a generic cake mix or a brand cake mix, that we can say, hey, that makes a great birthday party. So that's important in the context of I think what we're saying.
Thank you, Sam. Thanks, Markham. So a related question that we saw in the chat, I'll go to Dr. Liang for this one. How many batches of the products should typically be characterized? And I think a related question would be, sometimes maybe adequate number of batches are not available. What are the options in that situation?
Okay, yeah, thank you for the question. Actually, for this one, if there are published draft product specific guidances, you can refer to those PSGs. Typically, the recommendation is at least 3 batches. In cases that the RLD product is difficult to find, or the RLD is not manufacturing enough batch per year, you could use less than 3, but you need to provide a justification and also some supporting information to show that your effort of procuring these products, that you failed to procure so many batches, but you probably need to provide those kind of evidence along in your submission. So, thank you.
Thank you so much. I'm going to switch gears a little bit, and some of the questions that we receive for our talks from the industry, I'll go to maybe Samir first. We discussed a little bit about the impact of grade beyond like Q1, Q2 aspect of it, impact of grade on product quality or Q3. I was curious, like how often and for what kind of excipients we are typically seeing challenges where different grades are leading to differences in Q3 attributes.
Thank you for the question. I think specifically like polymer grades, let's say Carbamol or HPC, if we use different grades, have different molecular weights, it's difficult to characterize and identify, as Romit said. But if you use them at the same percentage what the RLD is using and a different grade, you may result in very different properties. So the ideal situation would be to actually determine what particular molecular weight is being used. And if you use that in the same percentage, you may get the same properties. So that would be one of the examples.
Thank you, Samir. A related question, and I'll go to Vaibhav for this one. Vaibhav, your video turned a little bit darker right now. Perfect. So I think in your talk, you mentioned the impact of age on Q3 properties for the RLD. Can you speak a little bit to how often this is being observed, and what do you do typically in these kinds of situations?
Basically, what I'll say is like we witness this quite often, specifically with cream formulations, and wherein like the consistency of product actually of the RLD product, it changes actually. And what we do is generally, as I said, like we will try to age match. Obviously, we cannot age match exactly because RLD is manufacturing at certain other time, and we are just when we are at a development stage. At that moment, suppose we have a 6-month or a 8-month development time now from the start of this development, and now we get a RLD, which is fresh one. So that may still coincide like viscosity or rheology, but certain times what happens is like when we start the development and when we take the RLD, so and those like in certain cases some products are like I believe RLD or the innovators, they are manufacturing them once a year or twice a year themselves. So we get one lot of RLD only per year. So we have to wait and then we have to collect that RLD and check the viscosity, rheology and the other parameters actually how. So what we were initially anticipating and how it is coming up now. It also happens when we go for an IVRT or an IVPT study. Our exhibit batches are quite fresh. We have prepared them and we have to go for a bioequivalent study. And then either the RLD turns quite old or we are getting a fresh lot of RLD, which is like too new for us to start with the bioequivalent study. So certain times there are so many nitty-gritties actually around this when we think of the age of RLD actually, and so certain times like we are just caught in this situation. So we have to, and most of the time like we cannot during the exhibit stage actually we cannot we are generally not evaluating it the RLDs to an extent that it changes into certain other things. So we have to keep it like maintain the integrity of the study as well, rather than changing anything in that scenario. So we are just caught in certain times actually with this. Generally, it doesn't happen with gel as much, but with creams, wherein like we have such polymers or like structure forming lipids or something are being used. So they tend to change their structure actually with time. So obviously the viscosity changes actually. And we also find like the structures, what I mentioned was, they are not visibly seen in bright light, but as you go with the polarized light, you can see those structures not as oil globules, but certain other structures. So you have to be very cautious while you do a microscopic test. We find it like people say it simply, but if you do and if you go into details, you can really find several things which add up actually to the product characterization, both from R&D perspective as well as from our development perspective as well.
Thank you, Vaibhav. Romit, would you like
Priyanka,
to?
Priyanka just wanted to add to that Samir's question actually. Sometimes what happens is like I have a polymer. So RLD is using a polymer, the same polymer I am using, but his polymer specification is on the lower side of viscosity example. And for us, we don't know actually what viscosity grade actually he is using, but the type of polymer, suppose carbomer 974P. So that is known, everyone is using the same thing. But if they have some specification design there at their manufacturing end, which says that, okay, you have, they will take that specific grade of carbomer that has a specific, that has a viscosity rate on the lower side of specification. So we may characterize the polymer as same, that okay, carbomer 974P, we are also using the same, but we may not exactly match the viscosity. So we may think that, okay, they may be using more amount of carbomer to yield the same viscosity. When we go with a more amount of polymer in that case, our Q and Q2 changes. So that also happens. So that way things are there actually. Yeah.
Thank you, Vaibhav. Romit, anything you would like to add based on your experience with the carbomers?
Yes, definitely, Priyanka. I think as correctly mentioned by both Samir and Vaibhav, some of the excipients as I presented, like very critical, and it's extremely difficult, like carbomers, to reverse engineer. We have tried to find out some of the ways on orthogonal techniques to identify, but still there are a lot of limitations that we are facing. Different grades has a different impact. Sometimes process has some impact on the product characteristics. And as correctly Vaibhav mentioned that, you know, when manufacturer has a range of viscosity on the specifications, and if we are not getting similar viscosity grades as an incoming material, there also finished product characteristics differs. and it will be extremely difficult for an industry or perspective to match those as a Q1, Q2. And I would like to add something more on, as I mentioned in our my presentation as well, right? Different grade of carbomer impacted a little bit different on pharmacokinetic characteristic as well, and we have to deviate from 5%. So that was a perfect example on carbomer. I want to add something more on Vaibhav's current observation on aging, right? So that specifically for some of the products which has multi-phase like creams, we have seen that there are sometimes limited availability on RLDs, and as the product ages, their microstructure changes. Some of the globule size that we observe with initial or freshly prepared product is different when we compare it against like 12-month-old RLD. So and there are not a lot of lots available. So in that case, it is extremely difficult for us to match those against our freshly prepared our product versus aged RLD products. So these are also some of the limitations industry is facing that we have to think about it.
Thank you, Romit. So I'm going to switch gears to some of our research-related questions. But before I do that, I did want to note that there were numerous questions in the chat about IVRT, IVPT, use of modeling and simulation for dermal products, and separately questions about ophthalmic products. I would like to encourage you to take a look at the CRCG websites, because we have had very, very extensive discussions on all of those topics starting August of last year. And those links, recording slides are all available on the website. Coming back to today's panel, the last, a couple of last questions that I have, and I'll start with Dr. Murthy. One of the questions that we seem to be getting in the chat is, when we are developing these in vitro-based methods, what is the impact of those application on the observed bioavailability results? And do we know what the correlation is between what we are doing in vitro and what we can expect to see in vivo when those applications may be different?
Thank you, Priyanka, for the question. I think it's a good question. I think this kind of a tool to evaluate the way the thermodynamic activity of drug changes, over the duration of IVPT study, and then correlating that with the performance of the product is still exploratory, I would say. I don't think there's any literature reports or any systematic investigation done to correlate this kind of the in vitro thermodynamic activity related data with the IVPT as such. But there is a lot of fundamental theory available correlating the thermodynamic activity with the permeation of drug. Dr. Richard Guy and Dr. Hadgraf and so many people have published on this, and based on that we have designed these experiments to take it further. Maybe eventually if we would be able to get to a stage where we would be able to predict whether the formulations would be bioavailable in the IVPT study, or whether the formulations are going to be therapeutically equivalent in the clinical study, just by looking at the way the drug, thermodynamic activity of drug changes in the formulation.
Thank you, Dr. Murthy. Yousuf, a related question for you. We are doing a lot of rheological assessments where we are, of course, using the rheometer for testing, and then we are doing in vivo studies. Do you have any additional thoughts on correlation between what we are observing in vitro to our in vivo data?
Of course, I think with the method development stage as well, our focus has been to try and develop methods that correlate to various stages of the product use, right from the time it's being manufactured and put into a container to in-use application stresses. So if you're looking at rheological behavior at all of these, and I think that only ends when you complete it with tribology, which is surface-to-surface contact. So if you include rheological assessment at multiple shear rates using thin film rheology, where you can go up to really high shear rates, plus do tribology, you probably cover everything that the product experiences in the use cycle.
Thank you, Yousuf. So we are at time and we will be wrapping up in a minute or so. One last question, circling back to Sam and perhaps Likan. What kind of criteria do we use for the Q3 stats? Do we need statistical criteria?
Likan, would you like to go first?
Sure. Thanks for the question. Actually, typically for Q3 tests, we do not need this kind of statistical criteria. However, for certain tests, that may be the case. I mean, this kind of criteria may be the case. I leave it to Sam to address that. So.
Thanks. So one of the things that you'll notice in the new batch of revised product-specific guidances that FDA recently released is that there's a very specific terminology that we use when referring to the in vitro tests that are used for Q3 characterization compared to the in vitro BE studies that are used for IVRT and IVPT. And with IVRT and IVPT, which are BE studies, there is a statistical assessment associated with the results. And the outcome is a dichotomous outcome that's dependent upon either pass or fail based upon a statistical assessment. So that's for IVRT and IVPT, which are classified as BE studies. But Q3 characterization is not a BE study, it's in vitro tests. And those tests are typically not utilizing a statistical assessment criteria. It uses more limit based criteria to make those assessments.
Thank you, Sam. Thank you so much to all of our speakers and panelists and to our entire audience who stayed back the extra 25 minutes to listen to our panel session today. Please enjoy your break and we will see you back promptly at 1:30 for a very exciting second session of this workshop. Thank you.
Thank you so much, Dr. Ghosh. Thank you, everybody.
Thank you.
Thank you.
Thank you, everyone.